Hello friends. So in our previous class, we talked about the metabolism of chylomicrons, which has to do with the exogenous lipoprotein pathway, where fats from our diet are ingested and transported to various peripheral tissues. But in this lecture, we are going to talk about the endogenous lipoprotein pathway, and that begins with the metabolism of very low density lipoprotein. So the hepatic cells, or the cells of the liver, function in synthesis of triglycerides, and that is called endogenous synthesis or de novo synthesis. Also, remember that when we talked about the metabolism of chylomicrons, we said that the chylomicron remnant was taken up by the liver, and then inside the liver it was hydrolyzed, or it will be hydrolyzed, and then the contained cholesterol esters will be released into the liver cells. Now, that cholesterol esters obtained from the hydrolysis of chylomicron remnant, together with the cholesterol synthesized by the liver cells and triglycerides synthesized by the liver cells, will be incorporated together with a protein called ApoB100, and this will form a very low density lipoprotein in the liver. Now, this very low density lipoprotein in the liver will be excreted or released into the circulation. On getting to the circulation, it is immature, and because it is immature, it is referred to as the nascent very low density lipoprotein. But then this nascent very low density lipoprotein will associate with high density lipoprotein, and high density lipoprotein has two apoproteins, ApoE and ApoC2. So this association will cause a transfer of ApoE and ApoC2 to the nascent VLDL, and that will result the nascent VLDL to become a mature VLDL. So the mature VLDL contains the ApoE, ApoC2, and ApoB100. Once this is mature, it will be carried by the circulation to various peripheral tissues, such as the adipose tissue, the skeletal muscle, and the cardiac muscle. In the capillary of these tissues, these various tissues, an enzyme called lipoprotein lipase is located on the wall of the capillary of these various tissues. So, on the wall of the capillary of the adipose tissue, cardiac muscle, and skeletal muscle, there is lipoprotein lipase. And then this mature VLDL passes through the capillaries. Now, on passing through the capillary, once it gets into the capillary, due to the presence of ApoC2, this enzyme, lipoprotein lipase, will be activated. So ApoC2 stimulates the activation of the lipoprotein lipase. And once this enzyme is being activated, it will hydrolyze triglyceride in this mature VLDL. It will hydrolyze triglycerides to form free fatty acid and glycerol. If this hydrolysis occurs in the capillary of the adipose tissue, the adipose tissue will absorb the free fatty acid and store it. But if it occurs in the cardiac and the skeletal muscle, the free fatty acids will be used to generate ATP, which can be used for the various functions that has to do with the cardiac muscle and the skeletal muscle. Also, the glycerol is water soluble, and because it is water soluble, it dissolves in the circulation or in the blood, and then it is taken up by the liver, where it can be used to synthesize glucose through a process of gluconeogenesis. When this triglyceride has been hydrolyzed, the major component of the particle, of the lipoprotein particle, will be cholesterol esters, still bound to the three apoproteins. But then again, HDL will associate with this particle. But this time around, its association is to take back the apoprotein which it gave initially. So here it gave C2, and then here it is back to take back the ApoC2. And then once ApoC2 has been transferred to HDL, it will result in the formation of intermediate density lipoprotein. Now, this intermediate density lipoprotein can do one of two things. It can either be taken up directly by the liver, or it will give off ApoE to form low density lipoprotein. So on the cells of the liver, there is a receptor that is specific for ApoE. So if ApoE binds to this receptor, then the ideal molecule will be taken up or will go into the liver through a process of receptor-mediated endocytosis. If the ideal particle is not taken up directly by the liver, then this ideal particle will associate with HDL. So HDL will again associate with ideal particle. And this time around, its association is as a result of the fact that it has to take back the ApoE that it donated initially. And now when it gives back the ApoE to the HDL particle, it will now result in the formation of low density lipoprotein. And the major fat in low density lipoprotein is cholesterol. But then there is a specific receptor for ApoB100 on the surface of the liver cells. So if the ideal is not taken up directly by the liver cells, it will give off ApoE and then result in the formation of LDL, which contains ApoB100. But then there is a receptor that is specific to ApoB100. So this ApoB100 will bind to its specific receptor on the liver cells, and then it will result in the uptake of the LDL particle. The major fat or the main fat in VLDL is triglycerides. So VLDL functions mainly in the transportation of triglycerides. However, there is cholesterol contained, but the amount of cholesterol is small compared to the amount of triglycerides. But when LDL particle is produced as a result of the hydrolysis of the triglycerides, then this LDL particle becomes rich in cholesterol. So VLDL itself contains mainly triglycerides and small amount of cholesterol. So in certain conditions, there is an imbalance between the synthesis of triglycerides and the synthesis and release of very low density lipoprotein. So ordinarily, as triglycerides are synthesized by the liver cells, VLDL will be formed. There will be incorporation of these fats together with ApoB100, and it will be released into the circulation. But then in certain conditions, more triglycerides are synthesized compared to the amount of very low density lipoprotein that is synthesized and secreted into the circulation. Such conditions include obesity, diabetes mellitus type 2, chronic alcohol consumption. So in these conditions, obesity, diabetes mellitus type 2, and chronic alcohol consumption, there is an imbalance between the synthesis of triglycerides and the synthesis and release of very low density lipoprotein. That is because more triglycerides are synthesized compared to the amount of VLDL that is synthesized and secreted into circulation. And that will result in oversynthesis and accumulation of triglycerides or fat in the liver, which is referred to as hepatic steatosis. So these various conditions can induce hepatic steatosis, where the amount of triglyceride that is synthesized by the liver cells overwhelms the amount of VLDL that is synthesized and released into the circulation. So basically, this entails the metabolism of very low density lipoprotein.